research level in genetics. Chita Kanabrian from LV Prasad, and does the same thing, a lot of research in molecular genetics. Dr. Natarajan, who was an excellent speaker. He will be with us a little bit later. He'll talk on retinitis pigmentosa, macular dystrophy, and diabetic retinopathy. And Sharola Damaraj, who goes between Moorfields Eye Hospital in London and Mangalore, where she does research too on non-ophthalmos Tay-Sachs and Leber congenital amaurosis. So you're in for an excellent panel of the best qualified American and Indian researchers in genetics. So hold on to your belts, here we go. Originally, genetics was limited to the esoteric publications of ORVO and ophthalmic genetics. However, I would say the general publications in ophthalmology and in other fields became interested in genetics and have highlighted it on their front pages. Here's the archives, and here's the Academy's issue of ophthalmology. Even the lay press has become interested in genetics. Newsweek, Time magazine. So as a result, we as physicians have to know genetics. Our patients know it, so we've got to catch up with them, because they're the ones asking us the questions. A lot has occurred in the Asian Games and the World Games. It was a question of what sex the players were, the winners. So what they did were testings. Notice the 46th for the number of chromosomes. But why pull down their pants when all you have to do is check their genes? And it gets a little complicated. We all know that the genes for maleness is on the Y chromosome, right on top, on the X chromosome here, which is not neutralized at all by the Y, which is much shorter. This is the area for maleness. And here's the Y chromosome. And if we get an exchange, it doesn't matter that much if it stays in the same body. But if it does change over and there's no counterpart, then the female will have the SRY gene and therefore have male characteristics. And here is the opposite with the male without the SRY gene. So even genetic analysis is not the answer. And even if everyone has all their chromosomes in the right place, there are other factors that could interfere with that. The adrenal glands producing too much testosterone, and so therefore it makes a female very masculine. So it's very confusing. And recently, in the newspapers, it gets even more confusing because we can see that if there are two in the mitochondria, in the right retina, and I'll show you in the cornea, two X chromosomes, one of them will be suppressed. Sometimes there's a mixture. In some cells, from the male, it will be suppressed. In other cells, from the female, it will be suppressed. So you have this battle going on within the women. And it shows up. Maybe I won't go into it, but I think this is one of the reasons why females differ from males. Because look at this battle. One is from the male, one is from the female. And here's the cornea. You can see the mixture in the corneal cells from the female X and the male X. So it gets very confusing. And here's the original article in Neuron. It just came out in January. People are interested in heredity. Am I going to pass on this? Am I going to pass on that to my offspring? What's going to happen to me in the future? Well, Time magazine wrote about it 2 years ago, and for decoding disease, and this is the complexity that they have. You can pick it up from Time magazine. All the areas that could be inherited. And recently, there's an article in Science Times in December of last year about the commercial aspects of having your genome analyzed. And there are companies out there. But turns out if you send your tissue to different companies, you'll get different results, different heredity patterns, different opinions. So much so that 23andMe had to shut down their genetic test. They were shut down by the U.S. Food and Drug Administration. We know about what happened with bin Laden and how they checked his genes. The reason I'm showing this, even though this was a couple of years ago, was that here in the lay press, it talks about DNA sequences and all that, 13 to 16 spots, tandem repeats. So this is what the people are reading. And right over here, it talks about short tandem repeats. Your patients are learning about it, and you've got to know it too. And this is the genome-wide association study, GYAS. We hope to pinpoint the changes in common disease, but it is disappointing because many of the diseases are not from just one gene, but the polygenic. And here we have certain disease, Leber's congenital amaurosis. Look at all the different genes that interact, and some of these genes can actually cause other dystrophies. And proteomics, it's the same thing. Catalog of the proteins in the cell, specified by the genome, making it even more complex. By the way, a lot of this is in your handouts. In the back, you'll see a glossary of terms, and you'll be able to know as much as your patients. We might touch on pre-implantation genetic diagnosis and in vitro fertilization. And, of course, we're worried about new viruses being made genetically. And, of course, I mentioned that genetic testing hopefully could predict a medical condition. Maybe we could have certain drugs specified for a certain patient, while another person with the same disease may not react to those drugs. And that's molecular therapeutics to reach a point of knowledge of a patient's genetics so that the medication be pinpointed. So, having introduced that and hopefully stimulated your desire to learn about genetics, I'll give you the crash course. It turns out there's only about 1 to 5% of the base pairs in the exons that code for proteins. The rest is called junk, but it really isn't junk. As a matter of fact, the New York Times said it isn't junk because it's used for hereditary issues, for family linkage. Now look, this is good. Differences in the genome from one person to the other is only about 0.1%. And over here, I want to just point this out. The unit of measurement, a centimorgan, about 1 million base pairs. It's used for measurement. So whole morgan means that for every meiosis, there will be a crossover. And you'll see that in a minute. But here the usual unit is a centimorgan, so it only occurs at crossover 1 out of 100 meiosis. Here we have the crossovers taking place. And so therefore the daughter cells in meiosis aren't pure from, let's say, the mother on one side, the father on the other side, but it's a mishmash, a mixture of both on each chromatid. Then we have it even more complicated, where we could get different chromosomes crossing over. Here's a mixture of 4 and 20. Nothing's easy. Anyway, when we talk about genetics, the center is the centromere. The bands go up in order, the numbers get higher, and on the Q, the longer, again, from the center down to the telomere, the numbers go higher. And here's the spread. We can see that the chromosomes get smaller and smaller and smaller, except that 22 is a little bit bigger than 21, X and Y. All these are wrapped up in histones, which are proteins, and all the histones are wrapped up like this, and then it's stuffed into your nucleus. Can you imagine? It is. Mitochondria. This is an important issue that's really of recent interest. Because, number one, your heredity is from your mother, not from your father. So any of the diseases which you'll see that are limited to mitochondria, you can blame your mother for. Anyway, mitochondria have to do with the energy, Krebs cycle, and also cell death. And here, the DNA is in a circle, and you could have a lot of mitochondrial DNA, each one sort of different, so you get a mosaic pattern. And of the number of genes, only 37 in the mitochondria, and only 13 code for proteins. Look at this. Ophthalmology has 3 of them: Leber's hereditary optic neuropathy, external ophthalmoplegia, chronic progressive external ophthalmoplegia, and Kern-Serre disease. Do you know that 90% of the genes that are express themselves in the tissue and cells of the eyes? I mean, the eye is a fantastic media for that, because any alteration, it's noticeable in the vision. So it doesn't take much for the vision to be affected. And that's why it's a good area for study. It's easily accessible, you can see it. Now here's mitochondria inside the cell, in the ribosomes, and all this take place. This is sort of a repeat. There are about 2 to 10 mitochondria in each of the cells, and many of these came from bacteria and ultimately became part of the nucleus. And here's the Krebs cycle in the mitochondria, and with fusion and fission creating the energy. All this is in your handouts, plus if you want to read more, the references also. And it even gets better. Let's say that there's a concern that a family has mitochondrial disease from the mother. So what do? They take the nucleus from the father, take a nucleus from the mother, in other words, the sperm from the father, nucleus from the mother's ovum. They then take it into another ovum from another woman who you know has good mitochondria and replace it. So here we have the outer mitochondria from a third person, the sperm, the egg nucleus from the parents, and if you want to, you could have it IVF'd into a third woman. So here's a child that could have 3 mothers. You talk about confusion? Okay, the breakdown. Everything is based upon 2 purines and 2 pyrimidines. That's the basis of all life, whether it's a leaf or whether it's a human. And we have deoxyribonucleotides and ribonucleotides all attaching to the pentose C2, and the base is on C1, and the phosphate C5. And here, this will be in your handouts. I'm not going to spend time on this. And here's the genetic pattern. We can see the male and female, and how it's passed on the generations. We go, this is the first, second, and third. And here is the person who introduced itself to the practice, and so it's marked accordingly, and those that are affected. This came from a study that Dr. Aldavi and I did on cornea plana. And this is very interesting because see that the mother has 2 partners, both of which produce children that had the cornea plana. But she has a third partner, and that one child doesn't have it. And we lost track of the fathers. They're in jail. Here's the purines. Notice that the color coded, and this is very important for the PCR tests. Polymerase chain reactions, that's being done in Dr. Aldavi's lab, and I'll just briefly show this. Here we can see how the purines and pyrimidines hooked up with the proline, and here, open reading frame, I think they're throwing me out, of how to read this from the second letter. All this, these are the amino acids, and we can see there are 20 amino acids, and each one can have various combinations of the bases. We have the start, the methionine, and we have the end, the stop codons. And here's the exons with the transcriptions into RNA, and we have the translation into amino acids, and again how you read it, and I won't go into this slide. And again, the open reading frame shift is when another, you could have a base inserted or deleted and it throws the whole reading off, and then you get a stop coding. If you have a missense, it's a change of one amino acid. And here's some associated genetic diseases when you have an elimination or a deletion. It's called nonsense. I won't go into this, but here's the article on genomics on the eye from the New England Journal of Medicine. The reference is in your handouts. Promoters and enhancers and silencers are all part of it. This is the encyclopedia of DNA elements that being referred to, and epigenetics is the study for hereditary changes other than evolution, which would be Darwin, and the draft. So I want to quickly go into, this is where the methylene tag is affected by environment, and that's epigenetics, and this is how the PCRs are done, the sequences, where we can see that one of the bases is changed for another because the color coding changes. Now it gets even more sophisticated, the chips and newer technologies. And we can see as a result, to check the genome, this is the cost involved. Now it's just over $5,000–6,000. Molecular therapeutics involve the blastocyst, where you could take some tissue, parthenogenic if it's just from the mother, and you can grow stem cells. You could even induce it from the skin, from fibroblasts. And this is a corneal orb that was put together, a combination from Sahara Nathalia, Dr. Krishna Kumar, and stem cell genetics in California. And here's the induced stem cells, transcription factors, where you can change from the skin fibrocysts into whatever tissue you want. And we can even produce the retina and part of the eye. For those that are more interested in this, there will be a course given in a couple of weeks here in India. Here's my references, and you can see that here we certainly are off to the future. Thank you.